Okay. Hi everyone, welcome to the CCMB seminar series. Today it's my pleasure to introduce Laurits Skov. Laurits is coming from the University of Copenhagen. He is at the Globe Institute in the section of molecular ecology and evolution. And before that, he was a postdoc at UC Berkeley in the molecular cell biology department.
Yes,
but
I think that's what it's called.
Also part of the Computational Biology Center at UC Berkeley. And before that, he did a postdoc at the Max Planck in Leipzig. And before that, he was a PhD student at University of Aarhus in Denmark. And today, he's gonna tell us about his research on archaic introgression. Welcome.
Thank you so much. Okay, so thanks to everyone for coming, and thanks for the introduction. So today I'm gonna be talking about these guys here. So this is a Neanderthal father and his daughter. And Neanderthals are extinct now, so they are no longer around. But in some sense, they're not completely extinct because their DNA lives on in us today. Because some ancestors met the Neanderthals and had children with them, so their DNA lives on in us today. And there's a lot of questions you can ask about this Neanderthal DNA. So I'm going to be focusing today on the more demographic history questions. So how many groups of archaic humans did we meet? Was it just Neanderthals and Denisovans? Is it maybe something else? And the question, when did we meet them? So I'm going to be focusing on the how many groups and when did it happen today. So before I start, just a little, so we're all on the same page. So today we are living in extraordinary times, in a sense that we are the only group of humans that are alive today. But if you went into a time machine and went back 100,000 years, there would have been at least 7 different groups of humans alive. So I try to illustrate this on this map here. So you have in gray, in Africa, early modern humans. So this would be the ancestors of everyone in this room. And then you have, and we think they are in Africa somewhere. So I just put it in the center. And then up in Europe and Western Asia, we have all the blue dots, and these are the Neanderthals. So it's another type of human group that were around. Then, if we go further to the east, we have the orange dots, and that's the Denisovans, primarily in East Asia, we think, and maybe South Asia. And then we have all these green dots, which are, so there's no DNA from any of these, but based on the morphology, that also looks like different types of humans. We have the Homo floresiensis, for instance, which is a very small type of human, which is called the Hobbit. So maybe you've heard about that one. And Homo naledi, which is the newest addition from South Africa. But, so what happened is that when humans left Africa, all these other groups disappear. Some of them disappear a bit before that. So they're no longer around. But because of the magic of ancient DNA, we've actually been able to sequence some of the genomes from these groups. So today we have some genomes from some Neanderthals, 3 Neanderthals to be exact, that are like good quality genomes, and 1 Denisova. And this was really thanks to many years of long work that eventually earned this guy, Svante Pääbo, earned him the Nobel Prize for getting DNA out of these Neanderthals and Denisovans. So this was a lot of work and a whole lot of money to get like these 4 ancient genomes. But with these genomes, we did also learn a lot of things. For instance, we were able to place these Neanderthals and Denisovans in our big family tree. And it's a little annoying with this zoom thing up here. Is there a way I can hide that? On the...
More outside. To the right.
That side.
Ah, thank you. Hide video panel.
Is that it?
No, it should say hide controls.
I can hide this one, maybe. No. Well, it's gonna be how it is. Oh, and a thing I didn't say at the beginning is that I timed this, so I think I have an hour, right?
Yes, but with questions?
Yes, but this is like shorter than that, so it's meant for you to ask questions as we go along. Is what I wanted to say. Well, so let me just see. Yes, I accidentally pressed one. So we have these genomes, and we managed to get DNA out of them. And then, you know, we can place it in a context. How are these groups of humans related to us? So what we found out is that something like 600,000 years ago is when our common ancestor lived. It was one population, and then there's a split. And one group becomes modern humans, if you will. So everyone who's like currently alive. And the other group becomes Neanderthals, who primarily lived in West Eurasia, and Denisovans primarily living in East Eurasia. But another thing that we also found out is that these groups met and they interacted with each other. So to be human is also to be a little bit Neanderthal and Denisova. And we found out that all people with non-sub-Saharan African ancestry trace something like 2% of the DNA to a Neanderthal-like population. And because it's in all non-Africans, we think that, you know, maybe when humans came out of Africa, they met Neanderthals somewhere close to that, and then spread out over the world. So maybe we think we met the Neanderthals in the Near East or something like that. Then there's also been evidence that there's been at least 2 meetings with this other group called the Denisovans. There is a big, there's a lot of Denisova, something like 1 to 3% in people in Papua New Guinea and Australia. And then there's also a little bit of Denisova that is a separate event in East Asians. So I try to draw these 2 circles here. But I really want to highlight that we're really not sure where these encounters took place. So all this comparing to the genomes and figuring out how much DNA they contributed, we can only do that because we have these ancient genomes. We have Neanderthals, and we can just compare where in the genome are you more like a Neanderthal or Denisova. But I told you earlier that there was like 7 different groups, at least, of humans that we know of. So there could be many more parts in our genomes that comes from these humans. So I quickly want to introduce, also because it's the computational department, like how we do this, how we find out which part in the genome is Neanderthal Denisovan, and I'm also going to show you how we can do it without even using those genomes. So I like to use this analogy that finding Neanderthal DNA in modern humans is a little bit like finding borrowed words in languages. So as an example here, I have 4 Nordic languages, and then I have English. And this is the same sentence in all languages. This is like, have a nice weekend. And what you might notice is that in Danish, weekend is a borrowed word from English. So we say weekend. So you can think of this weekend as being a Neanderthal integress piece of DNA. And English so then being the Neanderthal language, but don't worry about that. So, and this is what it would be in a DNA context. So we have different genomes, and some place this genome, the Neanderthal genome, is more like this genome, and then we can maybe say this part here comes from Neanderthal DNA. And this is all well and good if you have a reference genome, or if you know, for instance, in English that there's only borrowed words from English, then you could do this. The trouble is that in languages there's borrowed words from all kinds of other languages. So, and then in a genomic context, this might mean that there is, it's not just Neanderthals, it's also Denisovans, and it could be other groups too that contribute. And using this approach by comparing to a set donor language is going to bias us. So we want to move away from that, and we want to be able to figure out that this word is actually a borrowed word without knowing what languages come from. And I hope that you could maybe see that even without knowing that English is a language, you could maybe still appreciate that the word weekend is very different from the word helg in the other languages. So maybe there's some information there. And so this method has been my bread and butter for the past many years. So this is actually a method that does just that. So the idea is that you have three groups here. You have a group that has very little archaic admixture, or very little Neanderthal DNA, for instance. Then you have some group that, the target population here, so some group that did meet the archaic population and have some DNA from that. And then, of course, you have the archaic population. And then down here, there is a little piece of the genome, and each circle is a SNP, so a single nucleotide polymorphism. And the idea is that if you have a group that has very little archaic ancestry, you can just remove all the SNPs that are found in this group. And I'm going to show you here what that would look like. So if I remove all the SNPs that are in the out group, what happens is that there's sort of like two pieces of DNA left. One of them could have accumulated SNPs since the Out of Africa event, which is maybe, let's say, 50,000 years ago. And the other piece of DNA, this archaic DNA, has had something like 600,000 years, which is a split between humans and Neanderthals. So they had much longer time to accumulate mutations, because mutations sort of happen at a certain pace. So now this problem becomes, can you classify the genome into a region with a very low SNP density and very high SNP density? And so that is what this method is going to do. It's an HMM, so a hidden Markov model, and you just scan along the genome. You have some initial parameters, some transition and some emission parameters that you then train on each individual genome, and then you scan along the genome, and the model is going to say, this part of the genome has a very low SNP density, and this one has a very high. So the very high SNP density, I'm going to call it like archaic integrest. And that's all the method does. So it doesn't need to know what a Denisova or Neanderthal genome is, for instance. So that's sort of the tool we're going to be using here. Another thing that is a fact is that most genomes that we have today are of European ancestry. And so that's where the Neanderthals lived, but it's not where the Denisovans lived or all these other groups. So we are probably biasing ourselves a lot we can only learn about the archaic humans that contributed to European genomes. But so that's a very narrow approach. So in order sort of to do something about that, we decided to run this model on 4 different data sets. So we're using the most diverse data sets we can get our hands on. So it's HTTP, and it's 1,000 Genomes project. We're also using a lot of our standards, and then we're using this new data set from India, where it's 2,700 people who are like full genome sequenced 30x. And then we're going to try to see what we can learn about Neanderthals and Denisovans from looking for Neanderthal and Denisova DNA in those genomes. So there's a nice illustration here. So the idea is to use this HMM and scan the genome. And then we're also what we're gonna do is like, we want to learn about the Neanderthal and Denisova population. So everyone here maybe has a few percent of the genome, which is archaic integrest, so Neanderthal or Denisova, but it's not going to be the same 2%. So if you have a lot of people, if you have sequencing not enough genomes, you should in theory be able to piece together the entire Neanderthal genome, or the entire Denisova genome, or the entire genome of whatever other group our ancestors met that contributed to us. So we're going to see how far we can get with that a bit later. But first, there's going to be some summary statistics here. Like first, we just want to know how much Neanderthal and Denisova DNA is there in each genome. So when we do that, let's first look at Neanderthal, and I express it as a proportion, so the proportion of the genome that is Neanderthal, and each little bar here is one individual from a data set. So I like squish them together so you can see the name, so otherwise it will get very messy. But what you notice here is that the people with the most Neanderthal DNA is this green bar here, which is people from Oceania. Then the second most bar, the highest bar is East Asia, so there's this dark blue. And then it follows that, then it's South Asia, then it's the Americas, and then it's Europe. And then the Middle East, with the least amount of Neanderthal DNA per individual. Yes?
How should we think about the y-axis here? It looks like some of the lines are going above 2. So why does the y-axis go from 0 to more than 2?
Well, why?
It's the percent of it.
Yeah, it's just...
Percent.
Yeah. Ah, no, sorry, yeah.
I also thought it was 200%.
Yeah. Oh, no. Yeah, so yeah, but you see that. So it's a percent of your genome that is Neanderthal, and it sort of hovers between 1 to 2% using this method. Yeah.
Are you allowing heterozygous sites that are
So
in
this.
one allele Neanderthal and in one allele not?
Yes. So this is phased genomes. So we sort of consider each of the chromosomes separately.
And what is the granularity of the transitions? Is it single nucleotide or windows or?
Yeah. So this is windows of 1,000 base pair. That is like what a method can call. Yes.
Do you have any idea why there's such a high proportion in Oceania and East Asia when we mostly see Neanderthals or in Europe? Like, why is it so high over there when we don't think we were there?
I'm gonna get to that. That's a really good question, but there's like a build up to that, so I don't wanna spoil it yet. Yeah. Yes, but maybe you can still appreciate that Neanderthal is between 1 to 2%. There's not so much Neanderthal variation, or is there? That's also I'm gonna talk about that. Well, but maybe you can. It looks way more stark, the differences, if you look at Denisova ancestry. So again, the y-axis is still proportion in percent. But you see that there's definitely way more Denisova ancestry in Oceanian genomes, that's people from Papua New Guinea. Then you have sort of a tie between South Asia and East Asia, which hasn't been appreciated so much. Like most of the time, people talk about Denisova being more in East Asia, but it also seems to be in South Asia. And then you have less in Americas and less in Europe, and then even less in the Middle East. Yes?
How do you determine what is Neanderthal and Denisova?
Yeah, so we do use the genomes to compare them to. So this is so the model can find pieces in the genome where it says this is archaic, this is human, but you don't know what archaic. So here we are comparing to Neanderthals and Denisovans, and it's which genome do you share the most derived variants with? That's the one you're gonna be called as.
So are you only labeling it these 2 labels, or is there also like ambiguous archaic?
There's also ambiguous, and there's also none. Those were not so interesting, so maybe I can like already reveal that we didn't find any evidence of a new archaic, unfortunately. It just seems to be mainly Neanderthals and Denisovans. And so everyone else has every single genome we looked at has the same amount of not sharing SNPs with any of them. So we think this is mainly false positives. So there's no evidence to suggest that there is this unknown human that contributed a lot. Yes?
So like this is one Denisova genome, or is it more now? Or how many genomes are there?
So there is one high coverage Denisova genome. There's one in the works that's gonna get published soon, I think next year. Then there are a couple of very low coverage, highly contaminated Denisova genomes too. But I'm comparing to the one high quality genome here. Yes. So now that we have all these, we have sort of an idea of how much there is per individual. A fun thing could be to see, well, how much, now that we're doing this piecing together of all the puzzle pieces, how much of the Neanderthal genome and how much of the Denisovan genome can we assemble from this big data set? Like, can we actually go to get a full genome? And the answer is not really, but getting pretty close. So this is the entire genome. There's no Neanderthal or Denisovan integration on the Y chromosome or the mitochondria, so that's not shown here. But the rest of the genome is. And the way you read this is that every place it's orange is where there's at least one individual out of these 32,000 individuals we looked at has either Neanderthal or Denisova ancestry. So if it's orange, it just means that at least one person has it. And this is sort of like putting it all together. And where it's dark gray, that's the centromeres, that's the telomeres, that's regions of the genome we can't assess. So actually, and then where it's white is where there's no Neanderthal DNA. And these blue ones, these ones here, are the deserts, but they're a little bit special, so I'll talk about them in a little bit. Yes?
Sorry, did you say that the orange parts were where one person has at least?
At least one person.
Neanderthal or Denisovan?
Yes.
Oh.
Oh yeah, so this is both of them merged together. But I have the numbers here, so we are now up to 1.6 gigabases of Neanderthal genome, which is actually a little bit more than the sequenced Neanderthal genome. So we learn a little bit more about Neanderthals from our genomes than the sequenced Neanderthal genomes. For Denisova, we can reconstruct something like a gigabase, so a third of the integrating Denisova genome. And then there are some fun places here. So there are places where no one has any evidence of Neanderthal or Denisova ancestry in these regions here, these 4 blue ones that are highlighted here. So this has been known for a while that these were like at least very depleted. Oh, sorry.
How many individuals did you use to get this?
So this is something like 32,000. But 27,000 are people from Iceland. So that's there's not so much genetic diversity there. Yeah. So there's all these regions where it seemed that it was not a good thing to have either Denisova or Neanderthal ancestry because it was very quickly removed. And in a paper that's going to come out maybe also this year, maybe early next year, we looked at ancient DNA, and we see that these deserts form very quickly. They form almost immediately after humans and Neanderthals and Denisovans meet. So I think there is something in these regions that definitely that resist integration. But what exactly the mechanism is, I'm not sure of yet. Or then maybe another thing to highlight is, I know there's also a lot of lack of archaic ancestry on the X chromosome, which people have also speculated why that could be.
So how many years are introversions? Sorry, not introversions. Deserts.
The desert, so
So you're saying that you found 4 introversion deserts over like 30,000 individuals?
yeah.
So how long?
So they are a couple of gigabases each. I think they're like 10 gigabases on average. So they're quite large. Yeah.
The desert, how do you define, like, how do you know the difference between like a desert and just sort of something that's, I see some white pieces that maybe look quite as long.
Yes.
Is it like a size thing, or is it like what makes you say it's a desert?
Yes, so actually, like, maybe I was, you can get the full definition of a desert now. So it's not where no one has it, it's where you need to have, and this is how it's traditionally been done, so let me just explain that. So you need to have a megabase, and in this megabase you need the frequency to be less than 0.1% of Neanderthal ancestry. And then you can take this megabase and slide it 100 kb to the right. And then if that's also true, then you keep on going. and then you only keep those where you slide more than a megabase. That's sort of how you find it. It's very. Why did I pick that? Well, so this is sort of to be sort of trying to be consistent with the literature. So that's what the first papers that looked at this did in 2014, and there you go. That's how you do it. So I was part of another study where we did something else. We did like just complete absence, and you do find a lot of the same places, but you do find a lot of smaller ones too. For this paper, we decided to go with the stricter approach. But this is sort of like there's definitely regions with very little Neanderthal ancestry, and these are sort of like the most extreme cases that are not the X chromosome.
So you're kind of doing this joint Neanderthal desert thing. If you look at them separately, do you have like more? How much overlap do you have between the two types of desert? Because you might think it's deleterious allele, and one of them it wouldn't be shared, or maybe something like that.
So actually, like there is one desert, and I believe it's the desert on chromosome 3 that is in both of them, both Neanderthal and Denisova. In the other ones, you do find a little bit of Denisova here and there in the Neanderthal deserts, and vice versa. The problem, like, so the Neanderthal deserts, I think we're getting a good hang of now where they are. But the Denisova deserts, we're not having a good hang of. And the reason for that is because the amount of Denisova genomes is just way lower. So there's a lot of places in the genome where just by chance you have no Denisova DNA here. So this desert window sliding thing we're doing doesn't work very well. Yes?
So when you peek at those, because you're using HMMX, the modern humans don't have the high density of derived alleles, but do the archaics. Like if you were to look at that region in the Neanderthal, do you see a bunch of derived alleles, or do you also see ancestral alleles in both, right? Because your methods look at the density of private derived alleles.
Yeah. That are not seen in Sub-Saharan Africa. Actually, like that would be interesting to see what they are in these events. So that's not here, but that's actually very interesting. Like there is a method where they look for, because so this is not the first time humans and Neanderthals meet. We did meet 300,000 years ago too. And there humans gave Neanderthals the Y chromosome and the mitochondria, and also something like 5% in the genome. And it also seems that there are deserts forming there. So there's very little human integrase DNA into Neanderthals 300,000 years ago in these deserts here. That seems to be what's happening.
Right, but also there's like a high density of derived alleles in the archaics. Couldn't that just be explained by Genomical? Like those would be, if they were introgressed, they'd be purged quickly, because they would now be seen on the modern human background that has a higher effective population size, so selection would be a little bit more efficient than the archaic lineages that are like protected from a low effective population size.
Yeah.
If you were to believe they have low effective population size.
That's true, but why is it just these regions then? Because that seems to me like an argument for the genome as a whole. Everywhere in the genome, this would be true. But then you have it localized here. So I don't think that's the explanation. But I'm not sure. I'm not really sure what's going on here. I know some people have looked at that in these deserts, the gene density is slightly higher, but maybe more importantly, the recombination rate is lower, which could be a thing. But yeah, I'm not quite sure what's going on here. If someone has some good ideas, they would be much appreciated. What else do we have here? Yes. So I keep going back to this because I think this is going to tie into like the question about where we met Neanderthals. So you can sort of imagine that we make this plot. Like each time we have a new genome, like we count how much of Neanderthal genome could we piece together. So this is sort of what this is showing. So this is for people from Iceland, and this is where we sequenced 27,000 people. And so that's the x-axis, that each time you add a genome, how much of the Neanderthal or Denisova genome can you assemble? So you end up You see, like, there's some diminishing returns here, like you end up maybe getting at 1.2 gigabases of Neanderthal, and maybe like 0.1. But it's increasing more and more slowly, so there's probably no point in sequencing all 300,000 people in Iceland. You're not going to learn that much more about Neanderthals and Denisovans. But with this population from India, for example, is completely different. You learn way more by sequencing. There's 10 times more data in Iceland than there is in India, but you still learn way more about Neanderthals and about Denisovans. There's much more genetic diversity, which is true for Denisovans, but it's also true for Neanderthals. And actually, like, it doesn't stop there. It gets even more amazing. So what I'm going to show you now is sort of a Venn diagram, but presented in a different way. So this is called an upset plot. So what you have is the y-axis is how much. So like the first bar, for instance, is there's 300 megabases. And then if you follow the bar down, this tells you it's shared by who. So all the gray bars are things that are shared between multiple populations. So Neanderthal DNA that's shared between multiple populations. So the first one, for instance, is shared by everyone except Oceania. But what I want you to see here, and so another thing is that if I take all these bars and I sum them all up, so how much is gray, how much is blue, I get this Venn diagram here. And the colored bars is Neanderthal ancestry that is unique to somewhere. So the dark blue is Neanderthal ancestry that's unique to India. That's the name of the study that this is from. So that's Neanderthal ancestry that's unique to India. Then the light blue is Neanderthal ancestry that's unique to East Asia, and so on. So you can see here that most Neanderthal DNA in the world is shared, but the population with the most unique Neanderthal ancestry is India. And actually, if you were to add all the gray and the dark blue, you get that something like 90% of all Neanderthal ancestry seen in the world is seen in India. And I'm going to come back to that point. Let's finish this off by looking at Denisova ancestry too. So this is Neanderthal. Most things are shared.
So gray means it's shared by everyone.
No, so gray, if we take this one here, like this bar here, it's shared between all the populations that have a colored icon here. So it's not shared by Oceania, for instance. And the way you read this is like, this blue one here is not shared, it's only found in Lasida, or in India. So it's like a presence of absence of.
What makes a bar? Like, what is the X? It's sort of the tree. Oh, it's the treatments of sharing, I see.
Yeah, so it's like
looking at every combination.
Exactly.
Okay,
Yeah. And then I'm sorting them by how much, the abundance. Yeah. But each they are built up of these 100 KB windows that I'm just stacking on top of each other. So maybe you can appreciate that for Neanderthals, most Neanderthal DNA will be found in multiple groups. So most DNA is shared between multiple groups around the world. But if you look at Denisova DNA, it looks very different. Here you see that most Denisova DNA is sort of localized somewhere in the world. So we have this green bar here, which is Oceania, so most Denisova ancestry today is in Oceania. And also keep in mind that, for instance, the Indian data set is 2,700 people, Oceania is 28 people. So there's much more Denisova ancestry here, unique Denisova ancestry. But you also have a lot of Denisova ancestry in India. Those are by far the two highest populations. And this is also true if you downsample, so it's the same number of individuals in each region. Because here, like, they're vastly different. But if you downsample, that's still true. So all this unique Denisova and Neanderthal DNA in India sort of made us wonder, well, how can that be? Like, did they also meet Neanderthals in additional times, or what's going on here? And then there was also these other papers that came out. So this is not, I'm not an expert in this, but I'll try to summarize what these papers is about. So it's about finding ecological niches. So places where that you look at where has there historically been a lot of Neanderthal occupation. Okay, what was the climate like then at that point? And then you just like sort of extrapolate that to the entire world, like where would be a good place for Neanderthals to be? And you do the same with modern humans. And then what you find is that there seems to be this overlap in the mountains in Iran. So, why am I telling you this? So what I'm saying is that maybe this idea we had, like maybe the explanation for all this extra Denisova and Neanderthal ancestry in India is that that's where we met Neanderthals. That's where we met Denisovans. So maybe instead of having a model where we think that the ancestors of non-Africans came out of Africa and met Neanderthals in the Middle East, maybe it's like further east. Maybe this is actually where it all happened. And then from there, they spread into Europe, they spread into East Asia, they spread down to Papua New Guinea. So maybe you can like see the animation here. So that's food for thought. You can think about that. That's I think there's some evidence to support that. So that sort of wraps up how many groups did we meet. Does anyone have any questions before I go on to the next, more of a timing question? I'll take that as a continue, so I will. So this is a study that has been on the way for a couple of years. We're having some trouble getting it to work in all kinds of scenarios, and I'll tell you a bit more about that. But the idea is that we want to figure out when we met Neanderthals, for instance. And the way you typically do that is that you need to use some kind of clock, and people have traditionally used the recombination clock. So just a little example here, if you have the Neanderthal, the archaic population to the right, and then you have the modern human to the left, that is blue, and then Neanderthal is red. If you have a Neanderthal parent and a human parent, one of your chromosomes is going to be like completely Neanderthal, one is going to be completely human. But then as time goes on, recombination is going to mix these two chromosomes together. So the Neanderthal segments are going to be shorter and shorter and shorter. So if the shorter they are, the longer ago integration happened. So you could make a model where you can like take these short segments and you can do some math and some computation with those, and you can figure out when it happened. The problem is though that the shorter the segments get, the harder they are to find. So you're gonna bias your estimate. And so at some point it's gonna be invisible to us that there was ever a meeting between humans and Neanderthals. And another thing is that the recombination rate is different across the genome. So actually you need to go from base pairs to recombination distances. But for that you need good recombination maps, which actually don't exist for all human groups, because you need a lot of data to make them. So we thought about, is there maybe another clock we can use that's not the recombination clock? And we thought about, why don't we use the mutation clock instead? Because mutations also happen at a certain rate. So the idea here is that I have like a little diagram again with African population that doesn't have Neanderthal and non-African, and then the integrating Neanderthal and then a sequenced Neanderthal. So we're using the sequenced Neanderthal here. And the idea is that we can color SNPs based on where they happen. So if a SNP happened in a common ancestor of the Neanderthal population that we met and the sequenced Neanderthal, we can color it red. And then if it's unique to the integrating Neanderthal population, we can color it orange. And then the segment comes in to modern humans, but mutations keep on happening. And those mutations we call blue. So if we could figure out which mutations are which, we can simply just count how many blue mutations there are, and then divide by, well, how much sequence are we looking at? And that should give us the time of integration. So what turns out to be the case is that it is actually fairly easy to have a lot of data, that is, to distinguish these types of mutations. So this is data from Iceland, and I'm sort of showing you, yes? I'm gonna show you right now. Yes. So So this has to do with linguist disequilibrium, how often SNPs co-occur. So for instance, if you had a Neanderthal segment and they have like multiple SNPs in them, and you take two of these red SNPs, so SNPs that you also see in the Neanderthal genomes, and you say, what is the R squared between these SNPs? Then you get some number. Then you do that for all combinations of these red SNPs and segments, and you get a distribution that looks like this. So most of these red SNPs are very highly linked to each other. And that makes sense because that Neanderthal segment comes in at the same time, so all the SNPs are linked. Recombination doesn't have time to. And there's two other classes. So for these ones, they're easy because we know that Neanderthal and Denisova because we can just compare to the sequence genomes. But for the orange and the blues, they're not found in the sequence genome, so we can't compare them to that. They're just going to be absent. So what we're doing is that we're saying that these orange SNPs, they come into the population at the same time as the red ones. So if I did a R squared between the red and the orange, it's going to have the same distribution as the two reds. And that's what we saw empirically. So these two these are just things that are not seen in Neanderthal or Denisova genomes. So we have this bimodal distribution where some of them are very highly linked, and some of them are not linked at all. Does that sort of make sense?
So you think one of them is from the donor and
Yes, I think one of them came in at the same time as the red ones, and one of them happened after that. So they're not going to be linked. So this is sort of the idea. And you get this very nice bimodal distribution. And then what we did is we just pick some cutoff, and that cutoff is here at an R squared of 0.9. You could also pick another cutoff. It doesn't matter so much because there's not so much data there. So now what we do is this. Yes, we count how many blue SNPs there are, how many of these unlinked SNPs, and then we need to know what the mutation rate is per year, and we need to know how much sequence there was, and then we can get an estimate of the admixture time. Yes. So the next slide here is sort of like to try to convince you that maybe we have some good idea what the mutation rate is. So the y-axis is the mutation rate, and each little dot here is a study. So they are sorted by the year of publication. So these are some different studies that try to estimate what is the mutation rate in modern humans. And you can sort of see that it sort of hovers around this little gray box here. So now I'll just pick the mean in this gray box. And so that's going to be my mutation rate. And let's see what happens then. So if I use that mutation rate, there's a little spoiler in the title, but never mind that. So this is sort of the same where the y-axis is now trying to estimate the admixture time. So when did humans and Neanderthals meet each other? And the x-axis is different studies that have been trying to get at this question. When did humans and Neanderthals meet? And they're colored blue because they all use the recombination clock. But now we're going to try this mutation clock thing using just this point estimate that I told you before. And if we do that, we get something that's very consistent with the previous method. And a nice feature is also that it has smaller confidence intervals. So maybe we can actually get at the timing a bit better. So this is with Neanderthals. We really want to do it with Denisovans, but this only really works if you have a huge data set and you can accurately figure out what LD is between SNPs. So we haven't gotten it to work for Denisovans yet, but we're working on doing some tricks for that. So that was sort of that, and maybe like since it's more precise, you could say, oh, could you time the different events? Like was there multiple Neanderthal events or Denisova events, or could you like tell them apart? So that's sort of the goal here. We're not quite there yet, but I am getting there on time, so I would like to thank all the collaborators that helped me do this project. There's no way I could have done any of this all by myself. And then I also want to do a little advertisement because, as Emilia said, I am starting my own group in Copenhagen. So if you or anyone you know are looking to do a postdoc in Denmark, you can send me a message. And I have updated with my Copenhagen University email there. So thank you very much for listening to me. Yes.
So I know you were worried about the recombination clock because of short segments.
Yeah.
But have you thought about, or in coming up with this model, trying like a joint clock model, kind of like what they do, where it's essentially that you just it's still like a gamma distribution, but it takes information both from the mutational clock by looking at the differences with the sequence archaics, but you are using them anyways to identify who is who.
No, I like that. That could be a really good idea. Like initially, I wanted to have this. I like it when you take two different approaches but arrive at the same answer. So I wanted them to be independent initially. Maybe like, there's information in both of them that you could use to get maybe even more precise.
Yeah, because I remember reading that the mutation clock is better for dating older events, while the recombination clock has better clarity for more recent timing. And so I think there's a paper that advocates, you can use the best in both worlds in this joint clock model.
That's maybe actually a good idea, yeah, because now we're a bit stuck, we can't completely get it to work, but actually using the recombination might be a bit of a help.
Did you use only single nucleotide mutations for your mutation clock?
Yes.
And what is the evidence that the mutation rate was roughly constant over the past 50,000 years?
That's a good question.
Or not constant?
So actually, I went over this slide a bit quickly. Like you can see that there's different colors for the estimates, and that depends on how they were made. So some of them are de novo mutations, just taking two parents and counting how many mutations are in the child. So there will be this de novo mutation here. There's also some IPD, so you take some IPD segments and you see how many mutations are there. That gets you a few generations back, but it doesn't get you to 50,000 years. What does get you there is sort of this ancient DNA. So there's only one sample that's ancient DNA, that's this one here. And this is from an individual called Ust-Ishim, which is a 45,000-year-old woman, I think. No, maybe it was a man. But here you can say you know the radiocarbon date, so you know how, and you can take a modern genome and say how many differences is there between a modern genome and a chimpanzee, and then because it stopped accumulating mutations 45 years ago when the individual died, you can say how many missing mutations are there, and if you do that, you arrive at a mutation rate in the past 50,000 years. That is going to be this number, or 45,000 years, that's going to be this number. So to answer the question, like, there doesn't seem to be evidence that the mutation rate has been different in the past 50,000 years. And that's why we think we can use it.
And I guess my other question is, in your original method, how do you deal with large-scale inversions, like the well-known inversion of chromosome 17?
I don't. The answer is I don't. But because like what we're doing is that we're taking everything, we're mapping to a linear reference, and then we're only using SNPs. But you could do something where you use some of these pangenome graphs that are being generated, and maybe that wouldn't like actually help you with the inversion. No, I haven't thought about a good way to incorporate that.
So you're using a mutation rate like average across the genome, right?
I am not quite.
Okay,
so that also didn't explain that. So what we're doing is that we're trying to estimate also. So there is this average genome mutation rate, but we're trying to estimate in what regions is it a bit higher or a bit lower, and that becomes a factor that we scale with, trying to take that into account. But that's also opening up the can of worms that's assumed to be constant too over the past 50,000 years.
But I thought it was evidence that there was a slow mutation rate in humans. Whereas that compared to
No, I've been on a paper that also claims that. So, but the idea is that so there seems to be a higher mutation rate in Neanderthals and now also Denisovans than in modern humans. A 2% increase in those. And so there might have been a slowdown, but we think it happened after the split of humans and Neanderthals, but so close to that split.
So you use this mutation rate to sort of parameterize your model, but how sensitive is your like 50,000 years estimate to the choice of mutation rate?
It is gonna make it a bit vary a bit more.
Like you have this tight interval, but if you considered like across many samplings of mutation rates, would you get like
Yes. So
you
actually,
know how big it becomes? Like
yes. So the rate will be like, let's say I pick the middle of this gray bar. Like let's say we like sample and then we get to like the bottom of the bar and the low end of the bar.
yeah.
So that's gonna be something like less than 5% change,
Okay.
and a scale. So, and a scale like linearly here. So maybe 5% more, but so maybe you're getting up to here.
Okay, so it's still like
It's still fairly accurate, and this is with just using data from Iceland. So it's not the entire Neanderthal genome that we could assemble. So you also there's a bit more data to be gained. But there'll also be some uncertainty, as you say, like being more realistic about the mutation rate. Yeah.
Thank you so much.
Yeah, you're welcome.
Another cool paper that